epigenetics. Until recently, the aging process was considered to be an inexorable decline, characterized by the cumulative buildup of molecular damage to key cellular components, particularly our very DNA. Just as the various components of a car eventually break down over time, so do the components of our body. Challenging this assumption were life forms that could seemingly defy aging by attaining a sort of state of suspended animation, such as date pits unearthed during archaeological digs germinating after thousands of years, plants regenerating from fruits buried by Arctic squirrels 30,000 years before, and bacterial spores viable after tens of millions of years encased in amber or hundreds of millions of years preserved in salt crystals. However, one need not seek exotic examples to demonstrate the uncoupling of biological aging with chronological or calendar aging. Instances of the aging clock not only being halted but actively reversed and even reset to zero happen every day. The Great Reset. Think about it. A baby girl is born with all the eggs she will ever have. It may be decades before one of those eggs is fertilized. That egg could be sitting in her ovaries for 20, 30, 40 years, all the while aging just like every other cell in her body. Let's say she gets pregnant at 30. Upon fertilization, if that egg doesn't somehow rewind its aging clock to zero, then that 30-year-old egg could lead to the birth of another baby girl with ovaries that are then 30 years and 9 months old. By the time she gave birth decades later, the eggs would be more than 50 years old, and they would continue to age and accumulate molecular damage with every successive generation. This is why, necessarily, all manifestations of aging in egg cells must be erased. Otherwise, the eggs in women's ovaries would be millions of years old. In 1996, we learned that eggs aren't the only cells that can undergo a complete reversal of aging. That was the year a sheep named Dolly was born. The nucleus of an unfertilized egg was removed, and in its place the nucleus of an udder cell was inserted. Dolly is derived from a mammary gland cell, one of the key researchers said unabashedly while explaining her namesake, and we couldn't think of a more impressive pair of glands than Dolly Parton's, unquote. Then, with a little electric shock, the cell started dividing, no sperm required, and Dolly, the first animal cloned from an adult cell, was born. Previously, a frog had been cloned from a tadpole cell, earning the researcher the Nobel Prize, but Dolly was the first animal cloned from the cell of an adult. The world marveled that a genetically identical duplicate of an animal could be created. Since Dolly, thousands of clones have been made of mice, goats, pigs, rats, cows, horses, ferrets, wolves, deer, buffalo, camels, and dogs. Cats, too, the first one predictably named Copycat. The implications, however, go far beyond replicating particularly productive farm animals or generating Fido 2.0. Hidden within that one mature specialized cell dedicated to milk production taken from a sheep's udder was the full genetic blueprint for the entire animal we'd come to know as Dolly. Furthermore, the cell's age appeared to have been rewound back to zero. There's a lingering misconception that Dolly was beset by some sort of premature aging syndrome. After all, sheep lived to be about 12 years old. The mammary cell was taken from a 6-year-old, and Dolly died at age 6, suggesting that the aging clock had just kept ticking without resetting. But Dolly died from a viral illness, not old age, and subsequent experience shows clones can have normal lifespans. In fact, mice have been serially recloned, meaning there have been clones made from clones going out 25 generations, and they have all aged normally with respect to lifespan. So not only can adult cells be dialed back to an embryonic state, but they can effectively be rejuvenated by having any traces of aging wiped clean. Welcome to epigenetics. Genes load the gun, lifestyle pulls the trigger. The term epigenetics was coined in the 1940s before we even knew the physical nature of genes, a full decade before Watson and Crick and Wilkins and Franklin solved the structure of DNA. Epigenetics, which literally means above genetics, layers an extra level of information on top of the DNA sequence, which on its own is only about 750 megabytes of data encoding 50,000 genes. All our dividing cells are genetically identical, carrying a full complement of our DNA, but each cell doesn't need to express all of our tens of thousands of genes. Our nerve cells don't need to be pumping out liver enzymes, and our heart cells don't need to be growing hair. That's where epigenetics comes in. In effect, it's what switches genes on and off. There are a multitude of ways our body does this. I'll talk about sirtuins and microRNAs in their own chapters, but the best-known epigenetic regulator is DNA methylation. We have enzymes that can strategically add methyl groups directly onto our DNA to silence gene expression. A methyl group is a simple, stable configuration of carbon that can be added to flag stretches of DNA as skippable. It's one of more than a dozen ways DNA can be tagged. We have a separate set of enzymes that can remove these tags to turn the gene back on. There are approximately 28 million common methylation sites along our genetic code, most of which are methylated at any one time. The pattern of methylation is conserved when our cells divide, so a liver cell splits into two new liver cells rather than a bone or muscle cell, for example, and in this way, methylation patterns in sperm and eggs can be passed down through the generations. We used to think that once cells matured and had their DNA appropriately methylated to lock them into their specialized functions, that was it. But we now know that our epigenome, the pattern of methyl markings in our cells, is a dynamic system and responsive to external stimuli. Epigenetics allows organisms to more rapidly adapt to changing environmental conditions. It can take eons for large-scale shifts in the genetic code to happen, but the genes we already have can be switched on or off within a matter of hours. Epigenetics is how green grasshoppers can turn themselves black after a grassland fire to better camouflage against a charred soil, and how our body determines the number of active sweat glands we have in our skin based on whether we're born in the tropics or in a colder setting. Epigenetics is good news. It means our DNA is not our destiny. No matter our family history, the lifestyle choices we make can effectively turn on and off some of our genes, not only affecting us individually, but also our kids and maybe even our grandkids. In the Gene Expression Modulation by Intervention with Nutrition and Lifestyle, or GEMINAL study, Dr. Dean Ornish and colleagues took tissue biopsies before and after subjects adopted intensive lifestyle changes for 3 months that included a whole food plant-based diet. Beneficial changes in gene expression were noted for 500 different genes. The expression of disease-preventing genes was boosted, and the oncogenes that promote breast and prostate cancer, for example, were suppressed. No matter the genes we may have inherited from our parents, we can affect how those genes affect our health with what we eat and how we live. That's the power of epigenetics. Same DNA, but with different results. The most striking example of the epigenetic effect of diet on lifespan involves the humble honeybee. Queen bees and worker bees are genetically identical, yet queens may live for 3 years and lay up to 2,000 eggs a day, while worker bees may only live for 3 weeks and are functionally sterile. How can this be if there's no difference between them genetically? They have a different diet. When the hive's queen is dying, a larva is picked by nurse bees to be fed a secreted substance called royal jelly. The workers just get mostly a mixture of honey and pollen adorably named bee bread. When the chosen larva eats the royal jelly, the enzyme that had been silencing the expression of royal genes is turned off, and a new queen is born. The queen bee has the exact same genes as any of the worker bees, but simply because of what she ate, different genes are expressed, resulting in dramatic alterations to her life and lifespan, a 50-fold increase in longevity thanks to epigenetics. Sidebar: live like a queen? If royal jelly can turn a simple larva into a queen who can live more than 50 times longer, should we consider eating royal jelly ourselves? I review the available evidence in c.nf/royal jelly. Spoiler alert, while it may be the bee's knees for bees, given the rare cases of hemorrhagic or bloody colitis attributed to royal jelly supplements, eating PB and RJs could just end up being a royal pain in the butt. The epigenetic clock. There are certain DNA sites on our chromosomes that so predictably methylate or demethylate as we age that it's like clockwork, presenting a potential molecular crystal ball for human aging. In a remarkable triumph of big data, out of the millions of methylation sites in our DNA, a tiny subset so dependably shifts over time that you can predict someone's age within a few years just by strategically measuring the methylation pattern in a few hundred or even just a few dozen sites in someone's 3 billion-letter genome. Over the last few years, these epigenetic clocks have become established as robust measures of chronological age, surpassing telomere length, which I'll talk about later, as the best age predictor. Why invent some costly Rube Goldberg approach to divining someone's age when you can simply ask them? Well, yeah, you can imagine forensic applications, the determination of an unidentified victim's age with a blood or tissue sample, but that just scratches the surface. The kicker is that epigenetic clocks don't just track our chronological age, but appear to measure our true biological age. In other words, our epigenetic age can better predict our remaining life expectancy than our calendar age. Check out c.nf/clock for the whole wild story. It's like science fiction. Feed a drop of your blood in some futuristic machine that scans the placement of chemical markers on a strand of DNA, and out pops your true age, reflecting a lifetime of lifestyle choices. In addition to predicting time to death, epigenetic clocks also appear to foretell health span indicators such as cognitive decline, frailty, arthritis, and the progression of diseases like Alzheimer's and Parkinson's. As you can imagine, the insurance industry has jumped on this, and your premiums may soon be determined by your epigenetic age. But it's not some set-in-stone fortune-teller's curse. You can change the rate at which you age, and may soon be able to use epigenetic clocks to track your progress, potentially presenting a radically faster and cheaper way to test anti-aging interventions. Speeding and slowing biological aging. Studies of centenarians show that some age so slowly that a 105-year-old may have a DNA methylation age of a 60-year-old. No wonder they live so long. What can we do to decelerate our epigenetic clocks and slow down aging? Epigenetic clock analyses show that women age more slowly than men, which makes sense since women tend to live longer, a pattern so robust that one demographer quipped, "To be male is a genetic disease." To catch up, men may have to make even healthier changes to their diet and lifestyle. Cigarette smoke is linked to accelerated biological aging, with marked effects evident even at low levels of exposure. In contrast, both exercise frequency and intensity are associated with a deceleration of aging. What about meditation? 2 months of daily practice failed to significantly affect aging rates, and long-term meditators appear to have the same aging rates as non-meditating controls, though practitioners racking up an average of 6,000 hours of meditation may blunt the increase in epigenetic age acceleration over time. Until recently, caloric restriction had yet to be tested in people, but it had been shown to slow epigenetic aging in mice and monkeys. Over about a 15 to 21-year period of 30% dietary restriction, middle-aged rhesus monkeys appeared to epigenetically age 7 years less. Even more dramatically, mice at a 40% calorie restriction seemed to age only about 1 year over a period of about 3 years. In 2018, an aging analysis was published of the Calorie Study, the first major randomized trial of calorie restriction in humans. It's spelled Calerie. Using non-epigenetic estimates of biological aging, the control group continued to age at a rate of about 1 year per year, but in that time the dietary restriction group only seemed to age by about 1 month. And they achieved this with only a 12% calorie restriction, which is like skipping just one donut a day. Aging rates were slowed in the dietary restriction group independent of weight loss, but obesity has been associated with epigenetic age acceleration in samples of liver tissue and deep abdominal fat. However, even about 100 pounds of weight loss due to bariatric surgery did not appear to wind back the clock. Maybe we don't just have to eat less, but better. The lifestyle factor most associated with slowed aging, even more than exercise, is a marker of fruit and vegetable intake, blood levels of carotenoid phytonutrients like beta-carotene. So an epigenetic diet would focus on consuming more fruits and vegetables. On the other hand, the food most consistently linked to accelerating aging is meat. Perhaps this is partly due to the fact that blood levels of byproducts of banned pesticides like DDT are themselves associated with both accelerated aging and meat consumption. Long-term exposure to air pollution may also be associated with accelerated aging, but the data are mixed. Winding back the clock. The fact that our epigenetic age is a better predictor of lifespan and several diseases of old age than our chronological age is powerful evidence that DNA methylation is inexorably linked with some fundamental cause of age-related decline. Could it be what's actually driving human aging, or is it merely a passive marker of age? Is our epigenetic clock the cause of aging, or just the result? If it's an active driver, it's a driver that can go in reverse. Remember how, in cloning, an adult cell could be reprogrammed to revert it into an embryonic state? Not only were methylation marks erased to free up the whole genome, but all vestiges of aging appeared to vanish. We obviously don't want to reset the clock so far back that we dissolve into an amorphous blob, but might we be able to rewind the clock a little and rejuvenate our cells? In a Nobel Prize-winning discovery, stem cell researcher Shinya Yamanaka identified what we now refer to as Yamanaka factors, a small handful of DNA-binding proteins responsible for cellular reprogramming that serve in essence to return a cell to factory settings. With these tools in hand, an international team of researchers set out to turn back the clock by reestablishing the regenerative properties of nerve tissue. For example, young kids can actually regrow an entire amputated fingertip, bone and all, but we gradually lose such capacities as we age. The cells that make up the optic nerves that connect our eyes to our brains similarly lose their regenerative properties. With a little Yamanaka factor manipulation, however, the researchers were able to successfully reset the methylation marks to a more youthful state, restoring vision in old mice and rejuvenating human neurons in a petri dish. The cells seem to have retained a faithful copy of the epigenetic map from earlier in life that could serve as directions to reverse aging. Methylation calibration. Increasing our access to exercise, fruits, and vegetables while reducing our consumption of tobacco and meat may help slow aging, as evidenced by a deacceleration of the epigenetic clock. But what about directly changing DNA methylation? A lot of things affect methylation patterns, but the modifications are hard to interpret. For example, in one study, a high-fat diet caused widespread DNA methylation changes in men within just 5 days, affecting more than 6,000 genes that were able to only partially reverse 6 to 8 weeks later after the participants returned to their usual diets. And overeating saturated fat causes different methylation changes than overeating polyunsaturated fat. But to what effect? We don't know. Do the epigenetic changes play a role in the ensuing physiological effects, or are they incidental? We're just starting to tease out the consequences of the epigenetic changes induced by diet and lifestyle. We know now, for instance, that among the consistent methylation differences in vegans compared to omnivores is hypomethylation, less methylation of a tumor suppressor gene and a gene that encodes a DNA repair enzyme. Since methylation silences genes, unmuzzling them may help account for the lower overall cancer rates among those eating plant-based diets. Similarly, in vegetarians, the superoxide dismutase enzyme is less often methylated. That's an antioxidant enzyme that can squelch 1 million free radicals per second. The hypomethylation is associated with a threefold increase in the expression of that detoxifying enzyme used to explain the, quote, higher protection against chronic diseases in vegetarians, unquote. Beyond tweaking the volume knob of individual genes, there's evidence that large-scale methylation shifts can have health and longevity implications. If you boost the enzyme that actually does the methylating in fruit flies, you can prolong their average lifespan by more than 50%. Suppress the enzyme and you cut short their lifespan. However, this strategy has yet to be shown to work in mammals. Human DNA methylation is much more complicated, but the fruit fly findings suggest that increasing global methylation capacity may positively affect longevity. Turn over a new leaf. The nutritional factor most widely studied for its epigenetic effects is folic acid. That's the supplemental form of folate, a B vitamin concentrated in beans and greens that is converted into a methyl donor. Folate comes from the same word root as foliage, folium, the Latin word for leaf. The methyl group that ends up on your DNA may originate from the folate in your salad, for instance, or the folic acid in a supplement or enriched flour. The recommended daily allowance for most adults is 400 micrograms. Yet the average daily intake of older men and women is less than 300 micrograms, and a third don't even get 200. What are the epigenetic implications? Postmenopausal women have their folate levels moderately depleted by placing them on a relatively low folate diet to study the epigenetic effects. Even though folate levels didn't fall enough to show clinical signs of deficiency, such as anemia, within 2 months, the subjects suffered a genome-wide DNA hypomethylation across the board. It was reversed within 3 weeks, though, upon resuming healthy levels of folate intake. A subsequent study of even older individuals resulted in the same undermethylation, but took longer to reverse, underscoring the importance of maintaining adequate levels in the first place. Even without depletion, a meta-analysis of randomized controlled trials of folic acid supplementation using the most sophisticated methods of laboratory analysis found an increase in global methylation, suggesting that most of us may not be getting enough in our diet. There's no real benchmark for a normal level of methylation, so terms like hypomethylation are used in a relative sense, making these changes hard to interpret functionally. But our ancient ancestors ate a lot more leaves. They likely got twice as much folate as we do today, so the fact that our body puts the extra methyl group availability to use when we get a bump in folic acid levels suggests to me that our folate status may be suboptimal. This is easy to fix, though. For example, just meet my Daily Dozen recommendations for legumes and dark green leafy vegetables. Dr. Greger's Daily Dozen is available as a free app for iPhone and Android. A sidebar: hard as a MTHFR? So-called MTHFR mutations are a popular scapegoat often used by alternative medicine practitioners to prescribe special supplements that they not so coincidentally may also sell for a variety of common ailments. MTHFR is an enzyme our body makes to activate folate, a common variant of the MTHFR gene, which has a DNA code letter T rather than the more common C at the 677th position, makes for a less functional enzyme. This can have epigenetic implications, as those who got the T variants from both parents, about 10% of the global population, have diminished DNA methylation, but only when their folate intake is low. If you get enough folate, your methylation levels are the same regardless of whether you have the T variants. Similarly, those with 2 of the T variant genes may have higher risk of cancer, but again, it's only among those not getting enough folate. You don't need a special kind of folate either. The folate in foods and folic acid in supplements and enriched foods are perfectly usable, irrespective of which gene type you have. Since everyone should be striving to get enough folate, there's no benefit to routine genetic testing to see which variant you have, which is why major medical organizations in the field recommend against MTHFR testing. The only thing you might do differently if you knew you had a double dose of the less functional enzyme is to be especially careful about alcohol intake. Acetaldehyde, the breakdown product of alcohol, can destroy folate in our body, so those with double T variants should consider restricting their consumption to less than 1 drink a day. As everyone should probably be trying to minimize alcohol intake, I agree there's little value in knowing your MTHFR genetics. Folic acid is not the same as folate. A review of more than 100 meta-analyses of population studies show that those who get more folate in their diet tend to live longer and are protected against cardiovascular disease, several cancers, and a wide range of other chronic diseases. But some randomized controlled trials of folic acid supplements found increased cancer risk. As I explore in c.nf/folic, the mystery appears to have been solved when scientists figured out that we aren't rats. Natural folate isn't shelf-stable, but there's an enzyme in our liver that can convert the stable synthetic folic acid found in supplements into an active form of folate in our body. The original experiments were done on rats, though, and it turns out that their livers are 50 times more efficient at this conversion than ours. So we can end up with unmetabolized folic acid circulating throughout our body, which may impair our anti-cancer defenses. For example, randomized controlled trials have shown that men taking folic acid supplements significantly increase the risk of developing prostate cancer. Randomized trials have also found that those taking folic acid supplements for more than 3 years are more likely to develop colorectal polyps. So natural sources of folate, like beans and greens, may be best, though women who want to get pregnant are still advised to take folic acid supplements given their proven efficacy for reducing birth defects. Beyond food and supplements, the third way to improve your folate status is to contract out some of the production to your microbiome. A folate transporter in our colon appears to be specially designed to absorb folate produced by good bacteria like Bifidobacterium when we feed them fiber. Increasing your fiber intake can bolster the growth of little folate factories in your gut. Wrapping this up with food for thought, our epigenome, characterized by the pattern of DNA methylation, can be thought of as a lens through which our genetic information is filtered. Unfortunately, it's a lens that can become cloudy as it deteriorates with age. Thankfully, epigenetic changes are reversible, so we may be able to polish it back into focus. Caloric restriction, as well as diet and lifestyle improvements, including physical activity, smoking cessation, and shopping more in the produce aisle than at the meat counter, may all slow the epigenetic clock. Getting sufficient levels of methyl donor nutrients, such as folate, can also affect global methylation capacity. To help boost this anti-aging pathway on a daily basis, consider restricting calories by 12%, which would be cutting about 250 calories out of a 2,000-calorie-a-day diet, like skipping a piece of pie or cake every day. And meeting the 400 microgram recommended daily allowance of folate, which can be achieved with about a cup of cooked lentils or edamame, a cup and a half of cooked spinach or asparagus, or 2.5 cups of broccoli, for example.